welcome back, everybody. Thanks to Luke Rosen for kicking us off in the last session. Great job, and thank you for the great questions that came in. I think that it's really part of data DIY is to make sure your voice is heard throughout. And to continue the dialogue, we move into a panel discussion now that really it's data used for better clinical outcomes and creating standards of care. Our moderator, Liz Horn, is going to walk us through the narratives of kind of the different perspectives that help drive kind of movement towards better outcomes. And we'll talk about how patients and clinicians are using health data, including novel forms, to accelerate clinical impact, inform better outcomes, and gain insights into improving standards of care. So Liz, by all means,
awesome.
Take it away.
Thank you, Christian, and thank you everyone for joining us. And I want to thank Global Genes and CZI for hosting this. I'd like to thank everyone who's attending both in person and online. I know everyone's really busy and it's Friday, and I'm just so glad you're spending your day with us. So what I wanted to do today on this panel is first just we'll have everyone introduce themselves, but I wanted to give you just sort of an overview about how we're going to do it. So, oh, awesome. We have a pointer and a slide advancer. And so everyone will have a talk for a few moments, less than 10 minutes, and then we will have questions at the end. So in your packet, you have this lovely sheet that tells you how to ask the questions online. So you can always raise your hand, but you can also go to data.diy.cnf.io, and this is on this page in the folder. And so with that, I think I'd like to start turning it over to our panel. Sorry, I should introduce myself. I'm Liz Horn. I am a scientist by training. I'm a cancer biologist, and I've spent my career working with patient advocacy groups and nonprofit organizations to build research initiatives, mostly in the registry and biobank space.
I'm Luke Rosen, and let me tell you everything about KIF1A and my story. Just kidding.
I'm Scott Demarest. I'm a pediatric neurologist and epileptologist specializing in genetic causes of early life epilepsy out of Children's Hospital, Colorado.
I'm Ethan Perlstein. I'm a scientist by training, molecular biologist, geneticist, pharmacologist, and also biotech entrepreneur.
And I'm Steve Roberts. I'm the Chief Scientific Officer at the Tuberous Sclerosis Alliance, also a scientist and had a 16-year career in industry and research before these last 8 years at the Tuberous Sclerosis Alliance.
Great, and thank you all for being part of this panel. And I want to point something out. We all have diverse perspectives, and all of the people that are right here on the panel, all of the backgrounds, it's really important. So you need someone that understands the lived experience, the patient experience, the families, which we have with Luke. You need the physician that understands the clinical side. You need the basic scientist, because we've all gone to school for a really long time, but we don't treat patients. And so the clinician voice and perspective is really, really important. And I think what's also really great about Steve is just his industry experience in addition to being a scientist and then working in the advocacy community. So as you're starting to build these programs, you really need partnerships and you need good partners. And you have to look at the skill sets that people bring, and that's gonna help you be successful. And I think the industry perspective's a really vital one as well, as people start to want to commercialize things. So I'm going to turn it over to Luke, and really we're focusing this panel on better clinical outcomes and creating standards of care. And one of the huge challenges in rare diseases is, well, it's rare, and as you said, it might not be as rare as we think because people are underdiagnosed, but physicians don't often see many patients with the same condition, and so trying to find that expertise. And we obviously, for our families and for our patients, we need better clinical outcomes. So can you talk about the role that families play in improving this challenging situation?
Yeah, that's...
And here, you can have yours.
Oh, yes, that's the vital question is the role that families play is to communicate really what is meaningful, what a meaningful change would be from a therapeutic, what matters most to the families. Like I was was mentioning before. And I think that's really our responsibility in this whole process, is to make sure that, that clinical outcome measures are appropriate and the tools that are being used to measure them and to develop endpoints are actually clinically impactful. And you're not setting yourself up for failure within, clinical trial or the discovery process because we're not asking the right questions. So it's the responsibility of the foundations and the families to not just suggest, but to drive and make sure that the right questions are being asked. And if they're not being asked, just to scream from the mountaintop.
And I know you have some slides, but I wanted to ask a quick follow-up question. When do the families need to be involved?
Right away, right away.
Yeah.
It's at diagnosis, right? Because first of all, everybody has a different diagnostic odyssey or diagnostic experience, and that's incredibly important for progress to be made, right? And so if people are being engaged early, then you can understand, we have since, I was very lucky in that what I thought and what my wife and I thought was a horrific experience was actually, in comparison to other members of our community, quite easy. And by that, I mean, we waited 2 years to get a diagnosis and 4 months from the time we had our sequencing done, right, our testing done. I have since met families who are now members of our community that went 15 years going through these painful, unnecessary, expensive tests and that 4 months was excruciating for us. I can't imagine. I was just speaking with somebody who's been a parent who's been running headfirst into these problems for 10 years, and I couldn't imagine that. I can't imagine that. So I think that by understanding early, we can cut down on that painful diagnostic process. Answer?
Yeah. And then do you want to?
Oh, yes, and what can we play that video? So before we do, this is my, on what I think meaningful change is. This is one of the most, I'm so proud of my son for so many different reasons. He's a remarkable person. He's 7 going on 40. He's got a lot happening in his life, and this is not just a remarkable moment, but it shows what's truly meaningful, and it shows my daughter closing her fist and throwing something, which is progress, and it's also something that we've been trying to accomplish for a long, long time. And by controlling her seizures in a certain way, she was able to do this. And so I feel like, the scientific community understanding that might help, maybe. Thanks for playing.
First, you're gonna get a pen. Then you're gonna make a wish. And then you're gonna toss it into the water fountain and hope your wish comes true. And then, okay, no. Here, okay.
You can do an overhand, Suze. You got such a great pitching arm. Throw it like a ball.
You can't go. Here, want me to show you? So I'm making my wish. Oh, and don't tell your wish. Okay?
Yay! Good job, Suze. Good wish.
You want to make one more, Suze? Okay. Same way.
Yay! Nice. All right, guys, let's go. So, had we not adjusted her seizure medication, her tremor in her hand would have likely prevented her from being able to make that wish and throw that coin. And I think that telling the scientific community that it's really important for our kids to be able to make a wish and learn how to throw a coin into a fountain is beyond measure. And it's important for us to communicate that. And I won't talk too much because I've already said a lot of this, but we have such a heterogeneous condition, and that's a challenge in coming up with a standard of care. And we don't have a way of reversing the function of the gene right now, so everything is symptomatic management. A lot of our seizures are treatment resistant, so that's tricky. And you know, the kind of 4 pillars of things that we need to understand and also communicate to the entire community is that it's so hard to identify seizure type. I think Scott could probably speak to that better than I can, but you know, our kids are having debilitating status at night because a lot of the families aren't able to identify what is a seizure, and they're you know, you can manage seizures if it's done correctly, and we just need to have the knowledge to identify what is a seizure, and the progression of the disease, and how to understand. You know, one of the things we do is in that registry that I talked about, we also have a list of what anti-epileptic drugs each kid is on and which ones are working. And we can, you know, a family in Finland can understand that, you know, 60% of our kids take, you know, 2 drugs that are working together, and they can just, the clinician can make up his own mind as to whether or not that's right, but it's on the radar. And then we have, you know, the movement disorder is tricky. So this is really our basic standard of care, is just try to figure out, keep an eye on everything, and this progression of the, you know, the spasticity and the paraplegia, and try to come up with the right therapies, the physical therapies, the occupational therapies, and advocate for and make sure all the kids are getting the services that they need, because if we're not constantly working on speech and the speech therapy, the regression is going to be such that it's going to be beyond repair. But if we're constantly working on it and the kids are getting the right services and they're getting, you know, speech therapy 6 times a week instead of 2, that's really important.
So this actually might be a good time for Scott to jump in. Because from a physician's perspective, how do you see the role of patients and families, and how can it help you, and are there areas that can be challenging? And feel free to use your slides. The slides will be available, so I think we're going to be a little more freestyle here, since everyone will be able to follow up with them.
So there's a lot to say there.
Pick what you want to answer.
I think clearly there are challenges. Most of the slides I have are about sort of how do you guys maybe get physicians on your side? How do you get them on your team? And I'm happy to talk about that. That's more from an organizational perspective, but I think there's also how do you get the physician on your team on a personal level and in your own care. And I think one of the things that's challenging, you mentioned, you know, that we go to school for a really long time, and, you know, we try to know as much as we can about these things. I think most physicians really do have good things in their heart. They want to help. They are doing the best they can. The reality is, with 7,000 rare diseases that all look different, and every kid in that rare disease looking different than the next kid over, that's hard. There is a lack of literature to guide them, and I think in most cases they will take anything they can get as far as some guidance or some help. Not every physician will. I'm just going to be honest about that. But the ones that you want on your team will. Mm-hmm. So they're going to listen to you. They're going to be thoughtful about what you say and what is important to you. And they're going to deal with that. They're going to try their best to come up with a strategy with you to address those concerns. And I think if you're getting something less than that, then you have to do what you can to try to find that.
And can you talk a little bit about how to get the physician on your team? even from the organizational perspective, because on some level there's some strategies and some tactics that have been pretty successful.
So there's some introductory things, but this is the punchline of it. Doctors are just people. We're not any better than anyone else. We're not any worse than anyone else. We're just people living a crazy life like everyone else is. Find a doctor that's a little bit interested in what you have. It doesn't have to be a lot. It has to be a little bit interested in what you're concerned about, and foster that. Guidelines are great, but even some place that's willing to just see the patient and build expertise over time is a starting point for that. So as I kind of think about through these things, yes, we want doctors to be incredible and amazing. That's just not the reality. We're just people. We have all kinds of things that we're trying to juggle and deal with on a daily basis. This is particularly, I'm coming from a perspective in academic medicine, but I've got a boss, and my boss is telling me, you got to see this many patients, you got to do this, you got to publish paper, you got to do whatever, right? And it's a little bit different for different doctors depending on what their scenario is, but there are all these things that are being pushed onto their plate. And way down the bottom there, I really need to set up that database, right? Like, they may have the best of intentions to do that, but they may not have the ability, because at the end of the day, the dirty secret about academic medicine, as much as all the hospitals and the universities, I think I'm being recorded. This will probably come back to bite me.
You are being recorded.
Yeah. The dirty little secret, as much as we do want to help people, money. Money is the reality of the world. So you want your doctor to be smart, compassionate, creative. Creativity comes from the fact that we often don't have guidelines. We often don't have a playbook to follow. If we did, that would be easy and you could go to any doctor. But you need them to be engaged, and then you need to harness that engagement. And even a little bit of engagement is a starting place. It's a way of you getting your foot in the door with them. And then you build on it as much as you can. And the way you can build on it is by supporting that physician. You want this guy, right? He's engaged, he's interested. There's lots of things that we want to try to do in rare disease space. This is a list of a slew of different things. All of them require time, money, and effort. And at the end of the day, time and effort require money. This is just academic. It's not good or bad, it just is what it is, that I get to spend my time doing what I'm paid to do. And as much as I might love to spend all of my nights and weekends doing the things I'm not paid to do, in my particular case, I also have 4 kids at home and a wife who also works a job, and life is life. And there's only so much that any one person can do. So how do you get somebody to do what you want them to do? That's the unfortunate reality. You give them the support to be able to do that.
Well, and I think you bring up a good point, that we are all so incredibly busy, and we all have our lives, and we all have the stressors in it, just even from regular life, but then the other things that come up with illness or family members or just different challenging things. And you're right, doctors are people too. This is a really, really big list. It's important. Can you talk a little bit about, when you're working with a physician, not all physicians have done a lot in clinical research or on the research side. And for some physicians, some of this stuff can actually be really daunting. You say IRBs to some people, and you can just see the panic in their face. So can you talk a little bit about some of the challenges?
So I think that's an important element of this, and some of what happens to be important about why we have a panel here. Not all physicians come in the same flavors either, right? And so I happen to be a physician and a researcher. There's other people who are just physicians, and they're really not interested in research. You need them all on your team. The problem is, it's not just that you need one person on your team. You want whoever you can get on your team. That doesn't mean you're going to give money to anybody on the team, right? And I get that. You got to be smart about your resources, but you always want them engaged. So even if they're just caring for your kid, or even if they have no ability to do research, if you can get them to be willing to say, yes, I'll see these kids. I'll see all of the KIF1A kids that we have in our area. Send them to me. They're going to start to develop expertise. They're going to start to put together patterns. And that's a starting point for natural history. That's a starting point for guiding the kind of data and research that you want eventually, even if they're not going to be the person who puts the database together or the IRB or actually does the formal research. Because that is only going to be as good as the questions you know to ask in the first place. Right. And part of the problem with rare disease is that nobody's seen more than 5 kids with it. And it's really hard to know what are the right questions when you've only seen 5 kids and every kid looks different. Or adult, sorry, it's a habit.
Do you mostly treat kids?
Right, I
Okay.
treat kids, so.
Pediatric neurologist.
Pediatric neurologist, so anyway. But, so I think you get them on your team either way. You can help younger or less experienced research-focused clinicians also to connect up with other people as you build your network of people who are engaged. And then some of those other people can guide that process. The reality also of academic medicine is that the people who have all the experience and really know how to do everything have no time to do anything. They are the senior people that are running everything and have lots of stuff already going on that they're committed to. So having a junior person who hasn't done much research but is interested and engaged and enthusiastic, who is paired with somebody who can be a mentor to them, who doesn't have as much time, can actually be really powerful.
Yeah, and I think that when we're talking about skill sets, no one can be an expert in everything, and you need the people. I mean, you'll need a statistician on your team, and most of us, we understand a little bit about statistics, but we would never do the stats ourselves. It might be a good time to check in with Ethan. So as a basic scientist, the importance of early preclinical work, how does this all fit into what we've already been talking about?
Sure. So I've had the perspective over the last 5 years as CEO of a biotech company focused exclusively on rare genetic diseases to see what happens when you sort of watch the arc start with a family, go through what I think is the single most important piece of data that you have once you have a diagnosis, through a discovery process, which can wend and wind, and then if it's really successful and you're lucky, you might even have a clinical opportunity faster than you can imagine. So I just want to walk through that case study, if you will. So this is a family, the Carmichael family in Michigan. It's their daughter Maggie. This is at her 5th birthday a few months ago. She's got a rare genetic disease called PMM2 deficiency, or phosphomonomutase 2 deficiency. It's the most common congenital disorder of glycosylation, about 500 or so patients in the US, maybe 1,000 plus worldwide, but massively underdiagnosed. So everything obviously starts with a family and then with a diagnosis. And I'm probably sure most people in this room have seen a copy or something like this kind of text from a clinical diagnostic sequencing report. And I argue that this is the single most important piece of data you'll probably ever really have. I mean, this is what kind of starts off or kind of ends the diagnostic odyssey and caps off the cure odyssey. And increasingly, I think we're living in a moment where you can have a hyper-focused N of 1 cure odyssey, where you're really just trying to fix a problem of one patient. And that moment wasn't possible even a year or two ago, because I think a family now enters the arena with just so much at their fingertips that they can come up to speed. I mean, Luke was commenting about this, but like they can come up to speed so fast on so many issues. It's like blowing my mind now that I'm meeting this latest generation of families that are hot off the diagnostic odyssey and then just no time wasted and are putting together research plans like I've never seen. But it all starts with saying, what am I going to do with this particular mutation? Because our approach at Prolara was to make animal models, focusing usually on simpler animal models like yeast and worms and flies and fish, but it really is about making a patient avatar. These models have to be faithful as much as they can. They're going to always be imperfect, but they have to be as faithful as they can to the patient. And you can only do that by making what we call a patient avatar. And that starts with taking a specific mutation that is driving a patient's disease and engineering it into these models and then recapitulating the disease and then trying to fix it in the animals. So if you've seen this piece of paper and you don't know what to do, this is actually the moment you should be immediately activated. And this is basically those two pieces of information, E139K, R141H, that's really the two pieces of information you need to just unlock this entire cure odyssey. And so this is now showing you, okay, you've got a piece of data, and you can digitize that data, you can put that into a registry, you can swap it and share it and all that, but you have to turn that data into discovery. And this is showing that the protein that's deficient in Maggie in patients like her is an enzyme, and enzyme happens to come together like a dimer, actually more like this. And I'm just showing you, highlighting the different colors, the mutations that are affected. So Maggie's mutation is that orange residue, E139, at the bottom there, or at the top on the other protein, on the other monomer. And then R14H is that green one that's sort of right next to it. And so what we can do in the case of this gene, which is very, very ancient, so all animal cells, all animals and all cells in all animals require this gene at all times in development. So that makes it actually kind of tricky for gene therapy and some other approaches, but actually makes it really good for a small molecule, especially one that could be given orally and can get into the brain where you need to be for this disease. So this is showing you how one of the first steps where your data becomes something real in a discovery process. These proteins we can engineer to be expressed inside of yeast cells, and we can measure yeast growth the way that you can compare a bottle of Heineken and a bottle of Hefeweizen. Right? One's clear, one's got a bunch of yeast growing in it. Obviously there was yeast in the Heineken, you filtered it out, but the point is a simple measurement of yeast growing, and that's basically like, did they make a solution cloudy? And that's how you can tell there's yeast in there, and you can measure that, what we call cell density over time. And this is, I won't go through all the science of this, but this is basically what we've done is created these patient avatars in yeast with particular mutations, including Maggie's mutations. And then we're able to see how well they grow. And they usually have a growth defect, which is the yeast version of the disease. They're not very sophisticated animals, so they can't display all the phenotypes that a human would, but they still fundamentally have the same enzyme deficiency. And then you can ask, do drugs out there that we know of, or drugs that we want to discover, can they increase the growth of this yeast? And if they do that, we think that they might be doing that by boosting the actual deficient enzyme itself, the PMM2 enzyme. And that's something we actually were able to show. We could take patient fibroblasts and find out that a drug that was approved in Japan in 1994 for diabetic neuropathy, which is a very large indication if that were applied in the US, we found using yeast models and worm models and ultimately confirming in patient fibroblasts that apelrestat, this drug, increases the activity of the PMM2 enzyme about 30% above its baseline. It may not seem like a lot, but we think the transition from health to disease is a pretty steep transition in terms of you can kind of tolerate a loss of PMM2 function until a certain point you fall off a cliff and then you have the disease. So we don't think you need more than a little nudge maybe to get you back. And that's what we're going to be testing now. We can now work with Maggie's family. We found a clinician at Mayo Clinic who is interested in essentially doing an N-of-1 trial here. And we're right now filing the paperwork for the compassionate use IND. And this is a process that took, a couple of, 4 months, be 150 grand worth of sort of research cost. This is a best case scenario. We don't know if it's going to work in Maggie yet. We have all the data in vitro that says it will, but this to me is hopefully illustrating for you how you could take this one piece of data on a report. Probably you're not in a very good mood when you're reading that report, but then realize how quickly that can be turned into discovery and ultimately into something that may even be, through drug repurposing, something that could have a shot at the clinic. So my key takeaways are you can't embark on a cure odyssey without taking a first step. And you can kind of simplify things as mutation, models, medicine. And as I said, this is, I think, an N-of-1 moment we're in now where families who've always been highly motivated now have the benefit of, and they always had the internet, or at least for a while, but now there's just so much information at your fingertips. And not just about like any random gene, right? Because what if a couple of years ago you came along and you got the report that's KIF1A, and you do a PubMed search and it says 0, and you're like, oh, shit, nobody studied this gene. Well, with every month that goes by, the probability that your gene is an orphan is not true anymore. There's going to be someone who's at least published something about that gene. And then another year goes by, and then maybe they've solved some of the basic fundamental science problems that have prevented translation from happening. For example, I've got some friends, they're affected by a prion disease, and there was a while there that if you got diagnosed with that, and we were still figuring out what causes a prion disease, is it a virus? Is it a bad protein going crazy? How could you even think about translation if you haven't put a bow on that simple, basic, outstanding question? But again, with every month that goes by, there's just so much research output. And then at the same time, there's more and more of these experimental therapies. So if you have a dominant negative mutation, great, we'll give you an ASO, antisense oligonucleotide, and we can customize that. You've got a loss of function mutation, great, we can do X, Y, and Z. You've got a haploinsufficiency, great. And like a couple of years ago, that wasn't true that you could just whip up for any mutation an N-of-1 experimental, but still even something that was not just theory.
Well, and the technology, not only the technology, but also the computing power, everything's moving so rapidly. And the things that we can do now that we couldn't do even 5 years ago is pretty
But I just
phenomenal.
and that brings me to the last point, which is, yes, there's more computing power, that's good in specific applications, but I would say in general, this idea that somehow our artificial intelligence or computing is just going to save you is not true. What you need is what I call organic intelligence, which is a combination of the IQ, but also the EQ of all of the researchers, the advocates, the families. We're all bringing their own special skills to the table, contributing in kind. Because if you don't know anything about the gene, or very little is known, there's no way that AI or big data is going to do anything about it. You need to create data. You have to have little data before you have big data. And so we think that you best generate little data through organic intelligence, through this process of getting these all-star teams together and just attacking this problem as quickly as possible.
So a follow-up question, I've been thinking about getting families together with providers. I mean, that they're interacting in the clinical space. How do we get families and researchers together?
Yeah, I mean, I think that in one way is I'm sort of now, as I'm transitioning from the role at Prolar to sort of the next thing, there's a demand out there for CSOs and for advisors, someone who can give, or even CMOs for that matter. And I think that, I'm working on hopefully on one project which is about sort of an online platform for this particular community that would help matchmake researchers and families, especially when you still have open questions in the field that are preventing you from attacking the translational opportunities. But yeah, I think it's just mostly that, again, just more stuff that anybody is putting out there is just breadcrumbs that any family can find. And then they're really good at finding the breadcrumbs and doing the next thing. So
Right.
I think you can do some things to help make the matchmaking happen. But I think the matchmaking is just happening because you've got highly motivated individuals who are just, as Luke can say, are not going to stop until somebody picks up the phone.
Yeah, that's it's still a huge unmet need. If there was that matchmaking capability that you're talking about, I think that would be game changing, because the amount of, the amount of hours that we waste doing lit searches and going through every investigator who's an author and then trying to contact them and then not being able to, and then traveling somewhere, if there was that matchmaking capability would cut down on so much time.
I think from the scientist perspective, when you work at the bench, you work with bacteria, you might work with mice, but it's mostly cells and it's not people. And I think that most scientists really don't encounter people with the diseases that they work on. And I think that can be really empowering for them to see someone and hear the family story. So Steve, this might be a good time to transition to you, because you can talk from the organizational perspective, but also as a scientist that's really in the space.
Yeah. So I wanted to tell you a little bit about what we've done in terms of data collection and how it helps set the stage. I think one of the takeaways from Ethan's story is really chance favoring the prepared mind, or favoring the prepared rare disease community, because here was a drug that existed, but you had to have the pieces together. You had to have the genetics, you had to have the link to the patients, you had to have researcher doing an assay, and you had to have those tools available, right? Those chemical tools available. And so I think that's a bit of a story I can touch on from the Tuberous Sclerosis Alliance's point of view. Just to put in context in 15 seconds, tuberous sclerosis complex is a genetic disorder. It's characterized by the formation of tumors in various organs of the body, usually in the brain, the kidneys, the heart, the lungs, and specific parts of the skin. But also, because of disorganizations in the brain, epilepsy and autism are also very common in TSC. We estimate there are about 50,000 people in the US, so many of the disorders we've talked about are much smaller than that. But a really interesting, unfortunate yet interesting aspect of TSC is no two people are exactly alike. Even identical twins aren't affected identically with the disorder. So we may have 50,000 people, but we've got almost 50,000 phenotypes as well. But fortunately, many years ago, and I think you'll hear a bit about that in the next panel when we review our history a bit more, is where being prepared helped lead some advances in TSC. So the way that we collect clinical data is through our natural history database. This database was started in 2006, so it's in its 13th year. But I'd like to put in perspective, we were founded in 1974, so the organization was 32 years old before it started this. Very much unlike Luke's organization that started and then started collecting data. So times have changed. But the vision hasn't changed. It's really to better understand the progression of the disease, follow all the manifestations, which is a high bar, identify cohorts for clinical studies and clinical trials, and of course be a resource for helping as part of the infrastructure to help develop new therapies. We collect the data at 18 different clinical sites. We heard money is important because we're collecting from medical records. We have to support the time of somebody at those clinics to go take information from the medical records and put it in the database in a very standardized way, and that takes money. Over the years, perhaps I should have added up the amount, but it might scare people away. But it can be an expensive undertaking, yet very valuable. Right? We've had a number of uses of the data. Multiple publications. I pulled a few out. People have studied epilepsy treatment patterns. So we're capturing what drugs they've been treated with when, and you can see when that relates to when they had surgeries or did they need a second surgery. We can correlate, we can look at whether somebody had early onset seizures, so in the first year of life or even the first 6 months of life, versus those that developed them later, and how did that affect other aspects of the disease and outcomes. People have looked at correlations between genotype and phenotype. And then right now, I'll skip to the bottom one, we've been updating our clinical consensus guidelines, and one of our surgeons, brain surgeons, has been looking at data from our natural history database to inform the recommendations on how frequently monitoring needs to be done. Because without data, you don't really know. One doctor might do it every year, and one doctor might do it every 3 years. And both of them are doing what they think is best, given what they know in their experience. But it takes the data, and I think particularly from multiple centers, to be able to compare and say what really makes a difference or doesn't make a difference in the outcomes. And so this is just a diagram I created that really shows this link and this interaction between our TSC clinic system. So we have centers of excellence, if you will, or what we call TSC clinics that meet certain criteria. And by having those clinics and collecting data from a subset of them, from 18 of them, we have our natural history database that can help inform our clinical consensus guidelines, which then feeds back to how the clinics practice, which then affects the next, the subsequent data. And so we can continue to learn in this cycle. Currently, it's very labor intensive and burdensome to do that. And we get it from 18 clinics, but yet it's only 18 sites around the country, let alone the world. And so streamlining this process through technology would be a way to really, really accelerate and advance our understanding. So in my last slide, I just wanted to put this in context. So we've talked about helping stimulate therapies and advance new therapies and treatments. And so we see the natural history data as one piece of that, a very critical piece. And in fact, it's on the far right-hand side, in my mind, of the drug discovery and development pipeline, because it's documenting what the clinical aspects of the disease are. What is it that we're trying to impact through the development of new therapies? And so by beginning with the end in mind and understanding what we're shooting for, I think that, that helps set the path. But having that alone is perhaps not enough, right? And so we've added the collection of biosamples from people who were in there. So now you have not only the data, but if you, for example, if you go back to one of the previous findings and you say, well, there was a correlation between early onset seizures and negative outcomes, well then, can we look at biosamples taken from babies who were perhaps 3 months old and help predict which ones of those were going to have the early onset seizures and have the worst outcomes? So by coupling the samples with the data, then we're helping build those resources to get closer to the treatments. We have a clinical research consortium that has multiple sites that have worked together now on multiple studies, and we collect biosamples and natural history data from those sites. And then earlier in the pipeline, not only do we fund grants, but a really important program we put in place was our preclinical consortium to help get those data in our case, mice, but could be cells, could be whatever. And I think, Luke, I remember one of your slides from this morning about you wanted to make sure, you wanted to put in place a situation where companies couldn't say no to not work on something. Right. And that's really resonated. It's what we're trying to do, because we want to have the things in place that if a company has an idea for a potential molecule that they might have, that they look at this and they go, why would we not work in this? Because we have these pieces in research plus the connection to the community and the patients and the documentation of what they need and what's most important, and that's where we focus all of this.
And I think you bring up a really important point about biosamples, because we need the data, but we also need to understand the molecular basis. And before we open it up to questions, there was one thing that Luke said this morning that I thought was really, really important that I wanted to reiterate: the need to recontact, and the need to know who the data came from, who the samples came from, because if you can predict which babies are going to go on to have more seizures, if you can get a sample later, I mean, that's going to be so valuable. So, and it's really, there's a lot of planning that goes into this to make sure that you can do this when you get here, that you can do all of these things, but you have to set it up in the beginning. Recontact is so important. But before we open it up to questions, so I hope everyone's writing your questions down or putting them on the online website. I just wanted to give the panel a chance to ask questions of each other. Was there any follow-up or anything that we missed?
I wanted to emphasize something as well with this, and there's been a lot of discussion about why natural history studies are important, why that data matters. I mean, even from the slide that just disappeared, if you're waiting till that phase 3, if you're waiting to that phase 3 to do the natural history study, you're too late. Right? And so it takes time. It is a natural history study. It is longitudinal by nature. You have to be collecting that over a period of time, usually a longer one, not a shorter one. The earlier you start, the more ready you're going to be. And when we think about, I'm not on the pharmaceutical side, but I think I'm pretty comfortable saying that when you think about all the places you could invest, you're right, the treatments are coming. They really are. There's a slew, an armamentarium that is on the brink. And it's going to be invested in the places that are organized and ready to be able to measure whether or not it's effective. And we can't measure it unless we know what we're looking at. That's a natural history study. So if you don't have outcome measures, if you don't have longitudinal data, then that's not the group that they're going to invest in, because the same sort of treatments are going to be applicable to multiple different disorders. And I think the telling aspect of this is what's happened in spinal muscular atrophy. They can measure easily what happens with that disease, and it now is on the brink of 2 disease-modifying therapies, not just 1. It's actually cheaper to go back into the same disorder and try again with something. And that takes me to the next point, which I know everybody wants that first therapy, and I hope it comes very soon, but this will be iterative.
Right.
I'm sorry, but you're never going to get away from surveys. You're not going to get away from having to do the natural history data, because after we get the first one, there's going to be a newer treatment. There's going to be the next treatment in line, and this will be an iterative process. So the now treated patients become the new baseline for the new natural history that we have to demonstrate that there's an improvement above and beyond that with the second treatment. And this really will be how it will be. And I also apologize, that first treatment is not going to be perfect. Right? It's not going to fix everything. It's going to hopefully be substantial. It's hopefully going to give you the wins that matter to your community, or at least some of them. But we're going to be in a situation where we still have room for improvement.
Well, and it's a marathon, not a sprint.
Absolutely.
And I think you're being very pragmatic here, which is important. these projects take a long time, and there will be wins, there will be some setbacks, and I think perspective is important. I want to open it up to Pam, who's going to lead the Q&A.
Yeah, we have a comment from the audience.
I just have a quick comment. I just want to say thank you guys so much. I think I just wanted to reiterate a couple of points, and one of them was, you know, there's such a diverse group of patient advocates that are early in, like Luke, and that are from more sophisticated or organizations that have been at it with bigger patient communities and are farther downstream. But Scott, you brought up something so important. Not everyone can go out and raise a ton of money to, you know, pay someone to make all this happen. But what you showed with your slide about ways that you can contribute and that you can help be a participant, that slide that had all the different ways, the things that needed to be collected, that we can do as patients to help them be faster, more mobile, and entice them to become part of it is critical. And I just, please note, all of these slides are going to be available, but like I was looking at that thinking, wow, that's the list of here's how it's almost like money, right? Because you're taking the burden off you, we're managing that, and we can bring it to you for ideas, for your thoughts and comments, which get you back to your life on the weekends too, because it's unfair for us to think that you can do everything. And so I just want to say thank you for putting that up. I think it's so important to know that we can participate without always having to raise millions of dollars to really be drivers and help move these ships forward. And Luke has shown that with a lot of what he's done as well. So just thank you to all of you, because it was a really awesome panel.
Can I just say one thing about Steve and TSC? I joke with Steve and Kari a lot, and I say, you know, always stealing a page out of your book. But for advocates, and I'm very new at this, but one of the reasons I think that we're capable of making success is because we rip things off all the time.
And you should.
Yeah.
Because it's too hard to start from scratch.
Right. And like Nicole said, you know, groups that have been around longer and are more, I mean, that slide, that I'm quite literally going to steal a page out of this book here and take that slide back and say, guys, we need to implement this now. You know, so that's something to do, I think, is, yeah.
Thank you.
I want to challenge a little bit one of the comments from earlier. There was the question about how do we bring different disorders together and combine resources. And I think you're right that we need to pay attention to the differences between diseases and what makes them unique on a patient level and on a disease level. But I also think that there is a huge chunk of these diseases that the problems are common to all of them.
Yeah.
And I think there is actually an opportunity where organizations can come together to support common initiatives. 80% is a lot better than none, right?
Great
So
point.
that 20% might
absolutely.
be important about what's unique, but that 80% is a way that we can start to build as a group and be more efficient about how resources are utilized and spent. I think I could think of numerous ways, whether it comes down to infrastructure around how we collect data, around databases, around training the next generation of physicians and scientists, that every group could put in $5,000 and it would go a really, really long way, right? And so I think there are actually opportunities. It's just a matter of how does that organization happen on the patient advocacy side, because it's challenging on my side to say, can I get $1,000 from you and $1,000 from you and $1,000 from you to do this thing that I think is really important, that matters to all of you, but I can't spend the time just to organize it. And so I think there's opportunities there.
Here's a question from the audience.
I'm just going to comment on that. This is Megan O'Boyle from Phelan McDermott Syndrome Foundation. And that is actually happening, and it's exciting. And there are groups that do play well together in the sandbox. Two examples are Agenda, which I can't remember what the initials stand for, but essentially is genetic causes of neurodevelopmental disorders and autism. And Steve and Kari's group are part of it, and there's several other groups that are part of that. And we're going to go after grants together. We're going to study things that we all have as comorbidities. And we're also, a lot of us are sharing researchers. I mean, you know, Fragile X researchers are also Rett researchers sometimes. So It's to their benefit if we make that data more accessible to them, easier for them. Another great example is the Rare Epilepsy Network. Also, a lot of the same suspects that are in the other group, because a lot of people that have autism have epilepsy. And this is scalable. This isn't to say we have to lose our identities of our foundations. We can still be who we are. But whenever I talk to people about registries, I always say, go find your peeps, your cousins, the foundations that look like you, that have similar, symptoms, and go get their researchers interested in you, and go find out what questions they have in their registry, and rip it all off. But also, get on the phone once a month and say, we're having this problem with biomarkers. Have you all come up with an answer? Or, who else is having GI issues? Let's go to the NIH and try and get a workshop about GI issues, which is one of the things one of these groups has done. So I don't think we have to even just talk about it. I mean, this is scalable to, lysosomal diseases or dementias or, just pick a cohort. But the foundations need to make it happen. Nobody's going to knock on our door and say, we're having a meeting, everybody get together. We have to take the initiative.
Great, thank you, Megan. We have a question online, which is for Scott. What exactly makes an institution a center of excellence, and what can advocacy groups do to support creation of centers of excellence?
There's a better person to answer that in the audience, but and if she wants to, I'll let her speak. So I think I'm part of the CDKL5 Center of Excellence, and so I'm referring to Amanda Jackshaw from International Foundation for CDKL5 Research. I don't think there's any one answer. That's the first thing I would say. It partly depends where you are in the arc as a foundation and as a disease, the amount of knowledge that's there, amount of experience that's there. A center of excellence literally might be a guy saying, I'll see them. Just, yes, I'm willing to see those patients, and we'll try to figure things out. So it could be one doctor. A center of excellence might be a multidisciplinary clinic like the Rett Clinic at Children's Colorado, which has 14 different providers that see a patient in a single afternoon. There's different levels, and that's okay, and it will change over time. But I think it's sort of it's starting to say that there is somebody who's interested, and then funneling patients to them, because that's how it grows.
Yeah, I'll add that we found our Center of Excellence model with the Rett Clinic. So go find your friends that have similarities and figure out who are seeing your children, and go to the physicians that have an interest and at least a little bit of an idea of how to control epilepsy or whatever it be. Also, it's very difficult to take these models into different institutions and have them look exactly like they do at, Children's Colorado. So you have to be flexible, and you have to, we're really trying to define what kind of value these centers are bringing. So find friends, start small, and scale up. And I think there's big space for rare epilepsy clinics. I think there's a lot we can learn, all of us with genetic epilepsies coming together. So we started with Rett syndrome. We have a lot in common, but we also have early infantile epilepsy. So it doesn't make sense to just, stake our flag with Rett syndrome. We need to look around at other people who can also work with us molecularly and clinically. So, there's no guidebook. I wish I could say we could even put one together, but it's different anywhere you are, and I think that you have to really sit down with your community and figure out what's important to them and pull providers together and start there.
And if I could comment and maybe just reinforce the very last thing you were saying, which is when, ours kind of started, I don't know, 40, 50 years ago with Manny Gomez at Mayo Clinic as the person who was working on tuberous sclerosis complex. And now we have 64 clinics in the US that meet the criteria that we developed. So I think the thing that it could evolve over time. So it's critical to get started and have something. But as knowledge develops, then, you know, what you were saying, Amanda, is now the clinics are meeting the patient group's criteria, right? We can define what is it that we need to see, that our community needs to see from a clinic or a center of excellence, and redefine that. Because when you move the bar, not everybody moves up. So it can be an ongoing process.
Do we have another question in the audience? While I'm walking over there, I have a question for Ethan, and I guess these will be the last two. So do you believe that all rare diseases are caused by genetic mutations, and how long on average does it take to identify a mutation related to a specific disorder?
I mean, I think the stats are that 80% of rare diseases are genetic, but I don't know if I'm the expert to answer that. And then the second question was how
long on average would it take to identify?
Oh, how long does like a diagnostic odyssey last?
Yes.
I mean, I think the old stats were, you know, it could be 5 to 7 years, but I think with whole exome and whole genome diagnostic rates going up above 50%, I don't know what the exact numbers are. I don't know what that time is, but I imagine now that's, I mean, I think there's a lot of variance in that answer, because I can tell you families that have gotten an answer within a few weeks of birth. But I know that probably are still folks out there who are struggling for several years. I don't know what the average is.
Yeah, it's got to differ from situation to situation, and even where people are seen. If they're seen in I grew up in a rural place, so I'm not trying to be, you know, overly negative, but if you grow up or are seen in a place where there's not the sophistication, and the first thing they think of is not running a genetic test, right? It's everything else. The odyssey can go on for a long, long time. And then there are disorders where the gene's not known. And so even a genetic test is tough.
Okay, one last question.
It's more of a comment. Sorry to be blunt, but there's one word missing here when we speak about collaboration, is telemedicine. It is underutilized. You cannot practice rare disorders and expect people coming into clinic. You need the clinicians and the researchers to practice and advocate and perform telemedicine, because otherwise you stick to small numbers. Now, this is doable. It's, you know, you can do, there's a textbook how to do the guidelines, and it's not going to happen if you don't advocate for that.
Yeah, so Megan has a very good example of how telemedicine has really helped in their community.
Thank you, and I think this is very scalable and it's very exciting, and it took a parent who was new to our organization and had a child that had been very, very sick, and they finally got genetic testing after living with the diagnosis of autism for 15 years, and they found our foundation, and they were home, they found their family, and she started to hear similar stories on Facebook about what she had gone through with her daughter. And she's like, we can't wait 5 years for, the natural history study to be done and for publications to come out. We have to do something now. These families need help, and their doctors only have one patient, and they don't know where to go or what to do. So they used Echo. It's a free source, but you have to get training in it. And it's a telemedicine model. I really have to give credit to the generosity of our clinician researchers from around the country. They generously donate an hour a month to get on a video conference, and they consult with physicians or providers for any of our kids that are having issues. And my daughter actually, her psychiatrist went on and presented and said, these are the meds, this is what's happening, this is what happened. And they spent an hour with him. I didn't have to pull my kid out of school. I didn't have to take off work. I wasn't even there. And my psychiatrist was able to bill for it and get paid, because there's actually a billing code for something like this. And so he called me and he said, I got a laundry list of things we got to try. And again, this is scalable. Every rare disease could do something like this, but it really requires some amazing generosity by the physicians.
Can I just take that one step further, and how might be advice for some people? What we recently did, so we do something similar. I called Zoom, not promoting any company, but I called Zoom and asked Zoom, because you have to pay a certain amount of money to have Zoom conference. And Zoom gave us a subscription. So any community in the whole world, we can have one big Zoom call, we record it, and then make it totally accessible. And our clinician gets on the Zoom conference once a month, just like you, and we record it and share it with our entire group. So it's companies like, if you ask, I just asked. Well, I had to ask like 10 times.
Probably do the same thing with Slack if you wanted to have
Yeah,
like
with
Slack channels.
Slack too.
I'm sure they would.
They're
And I think
platforms
that's
to do
really
that.
important. I know we need to wrap up because lunch is next, which is exciting. But if you don't ask, no one will say yes.
Right.
So you have to ask. And I think people want to help. And I think there's just a part of that, just ask.
Great wrap up, Liz. Thank you. Thank you all very much. Christian?
Thanks again to the entire panel. Liz, great job also. And thank you to everybody asking these great questions from online as well and from the audience. I just want to give a little preview. Yes, lunch is coming up. 2 things I just want to mention though. Coming out of the break, which will be at about 12:30 Pacific time, 3:30 East Coast time. Data collection without a roadmap, how has it evolved? It's going to be a little bit more of the storytelling of the landmines found and the experiences that folks have been through from through their odyssey of building research, building collaboration. And then after that, the hows of data collection. This is where we start to really get into the playbook and the checklist items and the to-do list and the self-assessments that you filled out, and your readiness is where we start to really kind of get into the details. So right before we break for lunch, I just want, and this is especially for the live stream audience as well, we're actually, Global Genes is an organization that does a lot of different types of capacity building. This, data DIY is one of them. Another that's super important and core to who we are is making sure that our folks, our community, they know how to tell their story and communicate through different channels, through different mediums. And one of the reasons we have our own community members be such active participants in these programs is because they really do shine a light on doing it right. And it's trial by error a lot of time, but sometimes they know when they're doing it right and people can really learn from that. So I want to share a new video that KIF1A has developed sorry Luke, I'm going to do it.
It's fine.
That I think really shows that power of storytelling that attracts the clinicians. It keeps them motivated. It attracts more community members. And it personalizes the story that everybody's going through. And I just want to make sure that the folks online, I know it's lunch here, but there's something more to watch and keep it tuned in for this. So
I've only seen this once.
This will be my first time.
There were all of these little things that came together in a way that just was off.
We took her to the pediatrician, a lot of tests happening, and we're just trying to figure it out. They just were stumped.
And we took her to different doctors. We took her to, orthopedics, and then it was time, and then it was 18 months. And we had to go to the neurologist.
That's when things changed. I'm Luke Rosen, and I'm Susanna's father.
I'm Sally, Susanna's mom.
Susanna has KIF1A, associated neurological disorder, and what that is it's a very rare disease caused by a mutation in her KIF1A gene.
She has a movement disorder. She gets around differently, needs braces to help her walk, and she suffers from seizures.
Susanna's disease is not just rare, it's degenerative. So it gets trickier and trickier every single day.
There's nothing like the word degenerative to just pop any kind of bubble you had going.
It is just incredibly hard to be a parent in this situation. It feels sometimes like the weight of the world is on your shoulders.
Wendy Chung is our Sherpa,
I think of myself as a Sherpa.
helping us up this KIF1A mountain. On that day when Dr. Chung told us about Susanna's diagnosis, the question was, okay, well, how do we go find treatment? And Dr. Chung said, We got to find 100 kids, and we have to learn about the disease. And Sally and I made a decision to devote our lives to figuring it out.
I mean, Susanna's not just our child, she's our life's work.
2 years ago, while people thought there was just, 20 to 30 people with this disease in the world, we've since, just in 2 years, found, a couple of hundred.
One of the things we realize about KIF1A is that it's not nearly as rare as we might think it is. So we can see just over the past 3 years that we've really been watching the numbers grow.
Every day, a mom or a dad is reaching out and calling us and saying, hey, my kid just was diagnosed with this disease too, and we have similar problems.
It's a club that is one of the most amazing, caring, nurturing, supporting families, extended families that I know of.
I tell families when they call us and tell us that they've just been diagnosed with this disease, you have to contact our researchers and be involved in the research, otherwise our kids are going to die.
KIF1A has no treatment. It has no treatment, it has no cure, it has nothing other than symptomatic relief right now, and we know that left to its own devices, it will kill our children.
But now I wake up every single morning and my goal is to try to understand the drug development process and try to make it faster. We know that our kids with KIF1A have 3 sort of pillars of challenges: seizures, movement disorder, and optic nerve atrophy and vision problems.
KIF1A is a motor. It's like I think of it as a train going along train tracks. We literally see on a molecular basis a motor, the kinesin or the KIF1A, moving along the train tracks. Is the problem that it's going too slow? Is it a problem that it gets stuck? But we can use that now to be able to think about how we can get the train moving along again.
The research team, they're developing what's called a natural history study. And what that is you have to understand a picture of the disease.
Understanding the natural history, being able to see what the rate of decline is, if we can make any stabilization.
Understanding when those milestones get hit and when we start losing certain functions of the brain is really important for the researchers.
And incredibly important to get that right now, because at this point, even though we don't have a clinical trial, if we know in general that the slope is like this, and if I can have a medication that makes the slope like this, then to me that's a positive outcome.
It's hard to watch. Our kids' lives are so hard, and it's hard to watch things get worse. I'm never going to get to dance with my daughter at her wedding.
We are trying to buy time, and the best way for us to buy time is to do many things in parallel to try and just make them big in size so we can be short on time.
She is making herself our expert because we needed one.
The most amazing thing is that I don't do this alone at all. I do this with our families, and I do this with our scientific family as well. Incredibly talented people who are brilliant, but they are also some of the most dedicated, kind, loving people you've ever met.
This team of Wendy's, their work lives are dedicated to making our entire lives better.
With these rare diseases, there is no industry standard because they're not standard diseases, and so it's up to us and it's up to the families to work with the rest of the scientific community to create that industry standard.
We know we don't have an infinite amount of time to do this, and to be honest, these kiddos are our family. We have fallen in love with them. We have hugged them. We've played with them. They are our extended family. And we care about them just like we do our own children.
By having an understanding of both the science and the kid, I think, and I hope that that's the recipe for discovery. Yeah.
Nothing could be more important than finding treatment, and it has to happen now.
I knew I would start crying at some point today, so thanks.
I don't think you're the only one. It's okay. Don't worry about it. It's all good. Is it lunchtime?
Yes, it's lunchtime. It's lunchtime, thank you. And yeah, we'll be back in a few minutes. Thanks.